First of all, I'd like to thank the organization for giving me the opportunity to give this talk, which is a huge honor for me to be here as a PhD student. So I'm Pedro Aguilar-Garrido, and I'm working at the Hematological Malignancies Clinical Research Unit between the Spanish National Cancer Research Center and the Hospital 12 de Octubre in Madrid. So I'm going to talk a little bit about how RNA binding protein dysregulation could lead to a situation which is called bone marrow failure. So I have no potential conflict of interest. So as part of my research, we've got two different objectives. The first one is to study specific proteins, which are known as RBPs, and their implication in hematological malignancies. Some of these RBPs play a key role in the production of some of the hematological cells in a specific tissue known as bone marrow. So alterations in this type of molecules could lead to hematological malignancies. And in particular, we're going to study a protein which is called hnRNP. The second objective is to discover new targets to treat this type of hematological malignancies, such as hnRNP as a new target. So we know from our previous lab studies that hnRNP could act as a tumor suppressor or an oncogene, depending on the hematological malignancy. So finding inhibitors for hnRNP is a necessity for this type of pathologies. So we know that alterations in RBPs have an impact and could lead to a bone marrow failure. So we were wondering if alterations in hnRNP expression could lead to the same situation and pathologies. For that, we use cell models and mouse which overexpress this protein, trying to find possible alterations. And we found that there was an impaired protein generation and senescence-like phenotypes in our models. So what we found in our models, in cells, the overexpression of this protein leads to a situation which is called nucleolar stress, which is generated by an impair in protein generation and in ribosome biogenesis, leading to a senescence-like phenotypes. And in mouse, we've seen that hnRNP upregulation leads to an accelerated aging. And apart from all of the signs of aging, the bone marrow of these animals is not working properly. So there's an exhaustion of blood stem cells, and all the cell lineages are not properly generated. So in conclusions, we hypothesize that the upregulation of this protein leads to nucleolar stress, leads also to senescence-like phenotypes, and there's an increased protein production. So inside the cell, there's a lot of proteins which are not degraded well, leading to a toxic situation, which also contributes to generate this nucleolar stress. These alterations are commonly found in some diseases, such as myelodysplastic syndromes, or pathologies of the ribosome, such as the rarely Diamond-Blackfan anemia or Schwarzman-Diamond syndrome. So that's everything. Thank you, everyone, for listening to me.